So this time around, actually, we are going to look at the chemistry of amino acid and protein. So we are now going to start with the amino acids. So what is amino acids? Amino acids are compounds that contain an amino group, a carboxylic group, and a side chain. So whenever we talk about amino acids, the first thing that will come into your mind is actually the structure of the amino acids. And you can't talk about the structure of amino acid without looking at amino group, carboxylic group, and a side chain. And this side chain is a specific amino acid. So in an amino acids, we have carbon, and in this carbon, we have free hydrogen, and then we also have carboxylic group. We have carboxylic group, and then we also have amino group. And apart from these three groups attached to it, we also have R side chain, R side chain. And this R side chain is specific to amino acids. It is specific to amino acids. So this is how the structure of amino acids looks like. This is how it looks like. It has amino group, carboxylic group, and the hydrogen. There is free hydrogen. Amino group, this is the amino group, this is carboxylic group, and this is the free hydrogen, and then the R side chain. So in this structure of amino acids, as I said, we have this R side chain. So this R side chain is very, very important. It's very, very important because it is specific to amino acids. So amino acids have their R side chain as hydrogen. Some have CH3, that is methyl group, as their R side chain. Some have thiose group, that is SH group. So each amino group acid has its own specific R side chain. So it's not the same for all the amino acids. But amino group, carboxylic group, and hydrogens are present in all the amino acids. So they are present in all the amino acids. All amino acids contain carboxylic group, amino group, free hydrogen, as well as the R side chain. So an amino acids, and there are actually, we have 20 common amino acids. We have 20 common amino acids, and these 20 common amino acids are the ones that are used by the ribosome to synthesize proteins. Remember from our previous class, we actually discussed about the different functions of the cellular organelles. And one of the cellular organelles is ribosomes. So ribosomes is essential. It's like a protein manufacturing industry. It's an industry where we synthesize proteins. So what are the raw materials that are needed to synthesize proteins is amino acids. And these amino acids are 20. So we have 20 amino acids that are used in the synthesis of proteins by the ribosomes. And so also at the same time, these 20 amino acids have L-chirality. They have L-chirality. So one very important thing that we should understand generally, even from our basic chemistry, we know that there is what we call stereoisomerism. A stereoisomerism is a kind of isomerisms in which you have an alpha carbon. And what is alpha carbon? An alpha carbon is a carbon that have 4 different groups attached to it. If you have a carbon atoms attached to 4 different groups, so that carbon is called alpha carbon. Like for example now, look at this is alpha carbon. You see here we have carboxyl group, and this is amino group, and this is hydrogen, and this is side chain. So if you now look at this carbon, you observe that in this carbon, all the groups that attach to it are different. So that is why it is called alpha carbon. we call it alpha carbon. Or in the other way, we also call it, you can call it stereogenic carbon. You can call it stereogenic carbon, stereogenic carbon. So we have we call it stereogenic carbon, or in the other way, you can also call it chiral carbon. Chiral carbon. You can also call it asymmetric carbon. Asymmetric carbon. So all those isomerism, sorry, all those carbon atoms, any carbon atoms that have 4 different groups attached to it, we either call it alpha carbon, stereogenic carbon, chiral carbon, or asymmetric carbon. So all these names are all implied to the carbon that attach to 4 different groups. And we also have a way of representing compounds. That we have a ball and stick model. We also have amino acids. Amino acids have its own structure. So this is actually ball and stick model. This is a ball. You see, this is a ball, and this is the stick. So we call it ball and stick model. We call it ball and stick model. And then we also have the structure of the amino acid. How the structure of the amino acid looks like. It is tetrahedral in shape. It has tetrahedral structure. That is the shape of the amino acid. We have tetrahedral shape. Okay. So amino acids, they are actually the building blocks. They are the building blocks of proteins. It means, as mentioned previously, amino acids, they are the raw materials that are used actually in building proteins. So in proteins, we need amino acids. So each of these amino acids linked with one another to produce what we call polypeptides. They produce polypeptides. And polypeptides, these polypeptides are linked together by a bond, and the bond is called peptide bond. So peptide bond is a type of bond that linked one amino acid with one another. And usually a peptide bond involves the removal of small molecules. And in the case of the polypeptide formation, the small molecules that are released when one amino acid joined with another amino acid is water. It's water. So these kinds of reaction in chemistry, you call it condensation reaction. You call it condensation reaction, a reaction in which smaller molecules joined together to produce a larger one with the release of small molecules. It's called condensation reaction. Call it condensation reaction. So condensation reaction and polymerization are 2 different types of reaction, because in the case of polymerization reaction, of course, it's the smaller atoms that join together to produce the larger one, but there is no release of any small molecules. There is no release of any small molecules. So that kinds of reaction is called polymerization, but the one that actually involves the removal of smaller molecules is called, we call it condensation reaction. So most of the reactions that are taking place when you are actually trying to form the proteins is called condensation reaction. And there are 20 amino acids that are naturally incorporated into polypeptide, and they are also called proteogenic. They are proteogenic amino acid. When it is proteogenic amino acid, it means that they are amino acids that are used in the synthesis of protein. or in the other way, you can call it standard amino acids. So proteinogenic amino acid or standard amino acid are those amino acids that are involved in the protein synthesis. And these 20 amino acids are encoded by the universal genetic code. That is what you call a genetic code, which is a triplet codons that are used. Each of these triplet codons code for a specific amino acid. So these 20 amino acids have universal genetic code. So we have 10 standard amino acids, and this 10 standard amino acids, we have lysine, methionine, histidine, leucine, isoleucine, threonine, tryptophan, phenylalanine, valine, and arginine. All these, they are 10 standard amino acids, and they are called essential amino acids. And why are they called essential amino acids? Is because the human body cannot actually synthesize them. That is, we cannot create them, we cannot produce them. Rather, we actually obtain them from foods. So we usually get them from foods. Our body cannot synthesize them. So these kinds of amino acids are called essential amino acids. And here they are, which is lysine, methionine, histidine, leucine, isoleucine, threonine, tryptophan, phenylalanine, valine, and arginine. They are called essential amino acids. So all those amino acids that can be synthesized by the body are called non-essential amino acids. And those that cannot actually synthesize by the body are called non-essential amino acids. And then most of these proteins or most of these building blocks are used actually in the synthesis of varieties or different products. And these different products includes enzymes, hormones, antibodies, antibiotics, as well as the myriad of other substances having distinct biological activities. Remember, we have enzymes, and enzymes that are very, very essential in our body. In fact, remember that from our previous classes, we talked about that in human system, there are there are millions of reactions that are taking place in the body. In the body, there are millions of reactions that are taking place in the body, and these reactions are essential for the survival of living organisms. And most of these reactions are catalyzed by the enzymes. So, and these enzymes, they are proteins. They are proteins. Although it's not actually all the enzymes that are proteins, but at least more than 99.99% of the enzymes are proteins. So it means that they are synthesized from the amino acids. And of course, some of these enzymes are also, some of these amino acids are used in the synthesis of hormones. Although we have different types of hormones, we have peptide hormones, we have protein hormones, and we have steroid hormones. So peptide hormones and steroid hormones are peptide hormones and protein hormones. They are the hormones that actually synthesized by the amino acids. So we should understand that these amino acids, they are very, very important. And if you don't have them in the body, it means that there will be a problem. So if you are not taking, that is why actually the importance of taking proteins, because if you're not taking proteins, there is a likelihood you may have actually deficiency of some important biomolecules in our body. It may be enzymes, it may be hormones. And as I said, we have different types of hormones. Of course, what are hormones? Hormones are chemical messengers that brings about coordination. So the hormones, they are very, very essential in trying to maintain the homeostasis conditions of the body. We also have antibodies. What are antibodies? Remember, antibodies are proteins. They are proteins that are essential to fight the foreign antigen. Of course, remember from your basic knowledge of immunology, antigen is usually presented by a pathogen, while antibodies is our body that produce them in order to eliminate or fight back the antigen presented by the pathogens. So antibodies is also a protein. It's also a product of amino acids. Also antibiotics. What are antibiotics? Antibiotics, of course, are compounds or molecules that are used to kill microorganisms. So some of these antibiotics, they are proteins. It means that they are the products of the amino acids. So they have distinct biological activities in the body. They are also used as a single molecules in biochemical pathways. So some of these amino acids, they can be used as a starting or a raw material for a biochemical pathways. Like for example, if you want to synthesize melanin, the melanin synthesis starts with tyrosine. So therefore, tyrosine in the pathways of melanin biosynthesis, it's starting with tyrosine, and tyrosine is an amino acid. So it can be used as a single molecules in biochemical pathways. So now, what are actually the chemistry of the amino acids? So in this chemistry of amino acids, we have two functional groups. There are two functional groups that are found in the amino acids. And these two functional groups, there is carboxylic acid group, and this carboxylic group is represented as COOH. So we have this in an amino acids. Then we also have amino group. Amino group is also another class of functional groups that are found in amino acids. And it's also present on the alpha carbon. So we have amino group, which is NH3. So we have amino group. And then amino acids have different side groups. They have different side groups, and the properties that actually detect the behavior of amino acids. We have some amino acids that are acidic, some are basic, some are neutral. And that properties of the amino acid actually is due to the distinct nature of the amino group. It's due to the distinct nature of the R side change. It's due to the distinct nature of R side chain or the side groups. So each amino acid have its own R side change. And this R side change is actually very, very essential for the properties of each of the amino acids. So this is how it is. This is the amino acid. We have carboxylic group here. Then we have the amino group as presented previously from the previous slides. We have hydrogen here, and then we have the R side chain. This is carboxylic group, and this is the amino group, and this is the alpha carbon. Why is it that it is alpha carbon? Is because 4 different groups is attached to this carbon. So any carbon that have 4 different groups attached to it is called alpha carbon.
Sir,
Yes.
sir, please about that group.
Okay. What's the problem?
The different set of groups of amino acid. I miss it, sir. Please, can you re-explain it again?
Yes, what I'm saying is that in amino acids, there are 3 different groups that attach to the alpha carbon. And this alpha carbon are attached to 1 carboxylic group, amino group, and then the R side chain. And this R side chain varies or differ from one amino acid to another. And there is also free hydrogen. So, and then also I said, what is the meaning of the alpha carbon? The alpha carbon means it's a carbon that have 4 different groups attached to it. Like for example, now let's say we have carbon here, and we have hydrogen here. We have, let's say, OH. And then we have amino group here. And then we have another hydrogen here. So you see, this is a carboxylic group, this is the amino group, and this is the free hydrogen. But if you now look at this carbon, there is one hydrogen is attached to this, and there is also another hydrogen. So you see, this and this are the same. So it means that it's not 4 different groups that attach to this carbon. So since all the groups that attach to this carbon, they are not different. Sorry, they are actually they are not different from one another, because here you have one hydrogen and you also have another hydrogen. So it's not actually 4 different groups, because you have 2 hydrogen atoms attached to it. So this is not alpha carbon. It is not alpha carbon. But if, for example, now this hydrogen here is R. So you see, this is hydrogen and this is R. So this one is different from this, and this one is different from this. So they are all different that attach to this carbon. So it is called alpha carbon. Do you understand now?
Yes, sir, I understand.
And I also explained something like this on our previous class when we talk about carbohydrates. There is also a way of calculating the number of the stereoisomer, and the formula is 2 power n. If you want to calculate the number of stereoisomer, you can use this formula 2 power n. What is the meaning of this n? It means the number of the chiral carbon. That is the number of the alpha carbon. How many alpha carbons do we have in the compounds? Then you count them. Like, for example, here, we only have 1 alpha carbon. So since we only have 1 alpha carbon, then we are now going to say 2 power 1. So 2 power 1 is equal to what? 2. So therefore, we have only 2 stereoisomer. So it means that in this case, since we only have 2 stereoisomer, and this stereoisomer can either be either L or D isomer. So as I explained previously, most of the 20 amino acids that we have that are actually important in the synthesis of proteins in our body, they are in L form. They are in L form. They are in L form. And as we continue with the class, we'll see how the L and D differ from one another. So now, we are now going to look at the stereochemistry of amino acids. Stereochemistry of AAs, that is the stereochemistry of amino acids. So all amino acid except glycine are optically active. When we said optically active, it means that they have chiral carbon. They have chiral carbon. So now, how do we explain this? So we are starting, let's just look at L and D. So the 2 forms of enantiomers or stereoisomers. You see, this is alpha, sorry, this is L-glyceraldehyde, and this is D-glyceraldehyde. And what is the difference between these two? Okay, if we now look at L-glyceraldehyde and D-glyceraldehyde, you'll observe that this is the carbon. Look at the carbon 2 here. So you see that the groups that attach to this carbon, this one is different from this. And this one is hydrogen, and this one is OH. So it means that, and this one is alkyl functional group. So you see in this carbon and this carbon, you will see that there are 4 different groups attached to it. So since we have this is to tell us that a compound can undergo stereoisomer. It can undergo actually stereochemistry. And now let's look at what is the difference between L-glyceraldehyde and D-glyceraldehyde. It's just the orientation of OH group. It's just the orientation of OH group. And what is the orientation of the OH group? If you look at the L-glyceraldehyde, the OH group is by the left. And in the case of D-glyceraldehyde, the OH group is at the right. So you see D, it means dextrorotatory. Dextrorotatory. That is, dextro means right. Dextro means right. You see, this is right. Right. And then L lies laboratory. Labo means left. So it is at the left. It is at the left. So that is the difference between the L and D. Labo means left. Dextro means right. So we are just looking at the compound with reference to the position of OH group. Then now the next thing that we are going to look at is L-alanine and D-alanine. You see, here we are just looking at the amino group. So for the L-alanine is